Let me get my screen set up here. So you guys are going to see yourself initially, and then with my system set up properly, then you should see. Oh, it's giving away the punchline. Can you see that?
Yep.
Okay. Let me minimize yourselves. Get a laser pointer. Okay. So title of my talk today is Exploiting the Curse of Dimensionality for Metabolite Identification, and I'll explain that in a little bit. It's actually an interesting concept and argument in the field of mathematics. It has immediate application in measurement and separation sciences. So, so that'll be half of the presentation today. So and that's going to be very related to the metabolite identification probability manuscript that we've all been working on, in which you haven't heard anything about for a long time. But it's because we've been working up data to illustrate the concept of how this probability can work. And so it's taken a while to get the data. Then the other half will be sort of what we've accomplished over the 4 years of the program. And both of these things, the motivation behind what I'm going to present to you in our experimental computational approach for the PNA-CIC, both of these are driven from a motivation to come up with a method that allows unattended metabolomics, lipidomics data processing, and identification confidence assessment, all in a way that's machinable and ideally hands-off in terms of an analyst needing to look at spectra. And then the other thing too, Rick and I were talking earlier, we had 2 in-person meetings ever for this phase 2 in 2019, one in Bethesda, one at UC San Diego at Shankar's place. And it's just been a weird ride the last couple of years with COVID and all the virtual meetings. But I think we've done pretty well considering. So the curse of dimensionality was coined as a term by Richard Ernest Bellman, who was a applied mathematician. And this again was in the context of mathematical analysis and processing of data. It occurs in a variety of data scientists. And the important point for me is that when the principle behind this is that when dimensionality increases, the volume of the space increases so rapidly that the available data then becomes sparse. And so if you're trying to find patterns and or do relational analyses of data in a mathematical sense, the more dimensions that are considered, then the more difficult it is to do so. And so to obtain a reliable mathematical result, you need much, much more data in that multidimensional space than what you do in a lower dimensional space. So what this looks like in terms of math can be shown in 3 quick examples. The first is a one-dimensional view of data, where you might have data along a single vector, and say you've got 2 different data types in there, blue, green, and so you can look at it and see a pattern in the data. So it's sort of easy in that sense, but the clustering approach that you might use would be relatively naive there. Then if you go to 2 dimensions, you have the ability to better identify true patterns in the data. And then moving to a multi-dimensional space, you can get quite high accuracy in clustering, but in a mathematical sense, you have all of these caveats that come with moving into a multi-dimensional space, and particularly above or beyond 3 dimensions, where humans, they just can't visualize it or reconcile it in their minds, because we, of course, live in a 3-dimensional world, not in a beyond 3-dimensional world. So coming back to this point of when dimensionality increases, the volume of the space increases so rapidly that the data in that multi-dimensional space becomes sparse. This is exactly the concept that measurement and separation scientists have exploited for years. And we refer to this expanding multi-dimensional space either in single dimensions as resolution or peak capacity overall, or either in a single dimension or in multiple dimensions. And looking back at the seminal paper on reporting standards for compound identification, the highest level of identification confidence at that time was the MS level 1, which was a minimum of 2 independent and orthogonal data types relative to an authentic compound. And so right away, intuitively, analytical scientists knew that you couldn't get by with just a single dimension for highest identification confidence. You needed at least 2 in this example. And what I would hope to convince you of here is that more dimensions are better in compound identification. So I think of this again in the context of identification probability, and those of us that have been iterating on this paper have seen that. In probability, it won't be as ideal as a true estimate of false discovery, but I feel it will be a good first step. And also, all the methods that have been proposed so far for metabolite false discovery rate have all been in the context of a tandem mass spectra. They haven't included chromatography or NMR spectra or any of the other data types that we can measure, except for only maybe very recently with papers such as the all CCS paper and similar. Importantly, the probability will depend on the size and composition of the given reference library being used and the dimensionality and or resolution of the analytical platform being used, as well as the tolerances that one applies on each of the individual dimensions in that multidimensional analysis. And I always think of this in the context of the Hoberman sphere, where you've got a 3-dimensional space with very, very tight or very, very small overall resolution in the collapsed form. And then, yay, when you pull the thing apart, you've got a huge multi-dimensional space. And this kid could probably stick his whole head in there at some point and not bump into anything else in there. And so that's how I think of multi-dimensional metabolomics data filling that at least 3-dimensional space in this example. So I want to present to you now a toy example along with some data and ultimately hear what you think of it. So working with David Wishart's team at the University of Alberta, David is a co-lead of our computational core for SCIBC. We exported data for almost 2,000 molecules. We had their calculated mass to charge ratio, their predicted CCS, their predicted MS-MS spectra, and their predicted retention times using the retip approach and algorithm that was developed at UC Davis and Oliver's lab. And what I want to point out here is that all of this data is completely predictive. And so you will have molecules that will fit well within the training set in terms of chemistry and have good predictions. You'll have some that will be a little bit outside of the average prediction accuracies for any of these, but I think that's fine. There doesn't have to be completely accurate predictions, and the idea being that the predictions are a good enough representation of the distribution of the properties among chemistries, and so good enough for this example. So we exported all of this information and we set up a permutation matrix, and we varied the tolerances for these properties for some of them, not all of them, and we varied the combinations of properties that we considered, and then we matched the library to itself, and we did this multiple times. We define the probability of a correct match as 1 over n, where n is the number of matches to a given molecule within tolerances. And so, for example, matching just on mass alone, if we match within a plus or minus 5 ppm threshold, then n would be the number of molecules that fit within that threshold for a given target mass. And then a 1-to-1 match corresponds to a probability of 1, and additional spurious matches essentially penalize the probability of a correct match. So this is what the data looks like, and don't look too closely because we have trends falling on top of each other here. So I'm going to show a variety of individualized plots in a few seconds. But these are cumulative distributions of probabilities. And so the highest probability will be here, which corresponds to a probability of 1. And what that means is this is the number of molecules in that matching process per the number of dimensions considered and per the tolerances applied to those dimensions. These are the number of molecules that might have a match probability of 1 for those almost 2,000 molecules that were considered. So let me go now to the individualized plot so you can see a little bit more detail. So if we start out and consider only mass or mass to charge ratio, then you'll see we've got about 750 molecules with a match probability of 1, meaning we had 750 molecules which matched only to themselves when we matched the library to itself within the plus or minus 5 ppm window. And again, that's only for those 2,000 molecules that were considered. Then I've ordered these plots in progressing two-dimensional combinations in terms of the increase in molecules along the y-axis that achieve that same probability of 1. And you can see mass to charge and CCS here, CCS and MS2 spectra, mass to charge ratio and retention time, so on and so forth. And interestingly for me, at least in these two dimensions, combinations that involve retention time, such as mass to charge ratio and retention time and collision cross-section tend to give the biggest boost in terms of the number of molecules that have only a single match when matching the library to itself. And it's probably not too surprising since collision cross-section and mass to charge ratio, for example, aren't, they're semi-orthogonal at best. So moving to 3 dimensions and then 4 dimensions, you can see that the number of molecules with only a single match within tolerances increases. And we tend to maximize with these two comparisons here, one 3-dimensional, one 4-dimensional, at a little over 1,500 molecules. I think I estimated 1,600 here that have a identification probability of 1 based on how we defined it, which was about 80% of that 2,000 molecule set, compared to going back to mass alone, about 40%. And so this is a very first look at how this might work with, what you might call real data. Again, the CCS, the mass spectrum, the retention times are all predicted, but they're based on published and validated prediction tools, all which have some sort of uncertainty in the predictions. But again, I feel like the distributions are realistic enough within those predictive tools that this gives us an idea of how this approach to assessing identification confidence might work. And then in a more actual real data example, these are collision cross-section values versus mass to charge ratio from our IMS measurements of the unknown lipids working group data. Charles Evans gave a nice overview of that working group study. Sorry, I was just looking at time yesterday, and it's a fun group to work with, and it's been a tremendous amount of work, and it's going to be a great paper when it comes out and when we get there. But what I show here in dark blue circles are those features in our IMS data that align to consensus identifications from the working group based on either M over Z and or MS spectrum. And for the dark blue, that included those identifications that were confirmed by an MS2 spectra. And in the light blue, these are matches or identifications from the conventional analysis pipeline that were based only on a mass to charge ratio. And so they did not have accompanying MS2 spectra. And so what we've been able to do, and this is work from Caitlin Remford on our team, is develop these 99% confidence intervals and 99% prediction intervals to frame the lipid data, in this case for phosphatidylcholines. And you can see that there are a number of unknown lipids that did not have MS2 spectra, but were tentatively identified based on the mass to charge ratio that fall within the prediction interval. And so we would say those are probably phosphatidylcholines, and potentially be able to narrow it down at least to a summed carbon and unsaturation content. And then these dots that were features that fell outside of the confidence or prediction interval likely are not phosphatidylcholines and probably a misidentification based on mass to charge ratio alone. And you can see that for other lipid classes. These are diacylglycerols, these are triacylglycerols, and for some classes like this, we have things right on the edge. And so whether those are truly triacylglycerols or not, maybe they are, maybe our prediction intervals are a little too tight. It would still be good to get, I think, MS spectra there to confirm those, but certainly things that are far outside of this prediction interval very likely are not triacylglycerols. And then you can see that again for a single myelin class. So just to summarize the curse part of the presentation, I would propose that adding, and again, from an analytical perspective, I don't think anyone's going to argue that adding dimensionality, resolving things is going to not be a good thing. But from the toy example that we see, that we went through, it does look like adding dimensionality to measurements provides a higher identification probability, the way we defined it, by reducing conflicts in the matching process. The two caveats here, and there may be others that you can think of, and I'd be curious to hear about, but this is likely applicable only to known knowns, meaning you've got a library of known composition and you think these molecules are in your sample, or unknown knowns, meaning you've got a library of known composition and not all of those you think are in your sample, but you match the entire library against your data only. Where this will fail is if there are truly known unknowns or unknown unknowns in the sample, meaning they're not even in your reference library. So you could have a completely misidentification from that. And then again, those results that we showed are based only on predicted observables. But again, the more properties that we put together, the more dimensions of the analysis. Ideally, the less accurate one needs to be in any single dimension, because it's that aggregate of criteria that the identification needs to satisfy. So again, this was just based on 2,000 molecules. We know from proteomics that the more candidates you search for in your proteomics data, the higher the false discovery rate. And particularly for metaproteomic studies, for example, the microbial community and soil, the false discovery rate really goes through the roof if you include all potential genomes and proteomes in the search space. So one would have to be really, really careful here in terms of what they search for. And I personally would like to do a few other types of analyses, if possible, if time allows. But for the paper that some of us are involved in, I would at least like to go up to the full HMDB content, which would be a little over 200,000 molecules, and see what that looks like. And then in terms of MSI levels versus probabilities, I feel that the latter are highly machinable and interoperable between different dimensions of hyphenated measurements and or NMR and or UV spectra, so on and so forth. So I'd be curious to hear what folks think during the Q&A portion once we get there. And I would really be curious to hear if it makes sense and if you think you would use it. Okay, so now relatively quick review of what we've done over the last 4 years on the PNA CAC. So this is the conventional analytical pipeline for metabolomics right now. We identify things based on libraries that we've constructed through the analysis of pure compounds. And I think there's good momentum in the field right now to move to a different paradigm of relying on in silico predictions. And we've seen that from, I think, most, if not all of our CIDCs in terms of generation of tandem mass spectra via in silico means or CCS or even NMR spectra via in silico means. So I love it. I think it's a great trend. I think that will be the future, pending a lot of validation and just folks becoming comfortable with relying on that type of information. I already talked about the probability-based confidence score. And we also need, I think, really good software tools to bring together all of these different types of analytical data and via a matching process to arrive at a probability-based score, or ideally an FDR. So this is the new metabolomics paradigm that I envision, where rather than bottles of pure things, we have predictions of two-dimensional or three-dimensional chemical structures, or even ideally conformers, exactly how the molecule is shaped in a three-dimensional space. And that's how we framed our specific aims for the PNA CIC, predicting fragmentation spectra or collision cross-sections. Developing aim 2 is developing a multi-property matching approach, and then aim 3 was to disseminate all this back to the community. And we've done quite a bit of that. COVID definitely threw a wrench into the works for a variety of things, and we've suffered here from the Great Resignation, as I think have others, but we've either developed tools or used tools to predict CCS and MS-MS spectra. The tools that were developed under the context of this program were Icicle, which was our quantum chemistry-based approach for CCS prediction, and then we used DarkChem, which was developed under internal funding to, in grand scale, predict collision cross-section values. And these are all available on our website, and I think they're also now, most of these, if not all, available through HMDB. And the reason we settled on DarkChem was just because of computational time. Quantum chemistry is nice. It can be really, really accurate, but can also be really, really slow to achieve that level of accuracy. And then David Wishard's team has continually improved CFMID through the course of this program. And you can see some of the improvements here, looking at the CASME 2016 set. Also have developed tools to predict in silico metabolism. One of these was SciProduct, which focuses on biotransformations that are catalyzed by cytochrome P450 enzymes. That was published here. Further development or further dissemination of biotransformer via web server to get this capability into the hands of the community. That was just recently published. And also taking fairly detailed look at the effect of different molecular conformations in the gas phase and how those impact the collision cross-section calculations that are generated by Icicle as a representative quantum chemistry tool. That was also published last year. And you can see some of the data from that particular paper, how these different conformers affect CCS here, represented CCS versus energy. Again, we've developed the Icicle quantum chemistry pipeline. Again, David's team has continually improved CFMID, also publishing just recently a web server for CFMID 4.0. We've enjoyed looking at chemical space through a couple of different ways, published a paper on an approach for that. And we've developed a couple of tools either for standalone CCS extraction or for CCS extraction in addition to alignment and other processing of multidimensional mass spec data. That was just published a couple of months ago. And here's a cartoon that shows you the steps in the Damos pipeline for the different processing steps in that tool. Lastly, we've analyzed a lot of standards, and you're going to hear a little bit more about this tomorrow, in at least a portion of it in some lightning talks. Around 10,000 total molecules that we haven't quite analyzed them all. We've got another a set from the EPA that we have yet to analyze through the instrument, but all of this data is available, needs to be extracted, needs to be disseminated to the community, put on websites, web servers. And if you're interested, let us know, and we can disseminate all of this data to you for hosting on your own websites and so forth. So we already had 2 lightning talks. You heard those yesterday. I'll put in a plug for Jessica Bade here at P&L, who will talk about one of the things that has stymied us a little bit, and that is in processing ion mobility data, particularly when you're utilizing tandem mass spectrometry. The collision cell acts as another mini drift tube after the main drift tube, and so fragments will start to drift apart in that collision cell compared to the precursor ion as it exits the drift tube. And so we spent a lot of time reconciling that drift time offset, and Jess will tell you about that tomorrow. And also Christine at P&L will talk about an approach that we're taking to facilitate the automated processing of CCS values from one of these large sets of standards that we've analyzed. Last thing is we've had team members contribute to some best practice papers that have compiled with the working groups. Jamie led a nice one on a practical guide to metabolomic software development, and then Ryan Renslow at P&L really took this paper by the horns and got it out and published. Very good paper, very nice read on quantum chemistry calculations for metabolomics, and that was published in Chemical Reviews. So just to summarize, we've developed a variety of tools that will contribute to the development of reference library chemistries that we should be looking for, tools that will predict properties that can be measured, and then tools that will facilitate the extraction of the relevant data from these multidimensional analyses. Future work, since we're under no cost extension, we would like to continue to refine the quantum chemistry based tool Icicle. We would like to finish extraction of all of the data that we have available and to publish it and to make it available through websites and web servers. And really, I would like to operationalize multidimensional measurements. It doesn't necessarily have to be LC, IMS, MSMS, but some reasonably high throughput multidimensional platform that gives the ability to fully autonomize the data processing and include a machinable measure of either identification probability or of false discovery. And then, of course, disseminating and publishing the results. And with that, I'll thank the primary team members that you can see here. And also I would like to thank our consortium colleagues that we've been interacting with for the most part virtually over the last 2.5 or 3 years or so through working groups and other activities under the consortium. It's been a lot of fun. And I'll take any questions.
Thank you, Tom. So I can get questions started. So two things on the false discovery probability identification. So the first is, can you speculate a little on why adding the second dimension of fragmentation doesn't add much, as the fourth dimension over top of the parent ion CCS retention alone?
Yeah, it could be a dependency on the particular 2,000 molecules that were selected for generating the information, the predicted values. We didn't take a hard look at what those 2,000 molecules really are yet in terms of, say, a classification of molecular chemistry. The other thing that I did not mention is we selected a single collision energy range for predicting the MS-MS spectra. It was a medium collision energy, and so it may not have generated for those particular molecules informative enough fragmentation spectra that would allow some more specificity in the multidimensional matching process.
So then the second question, which I readily admit I was doing a little bit of email, so I may have missed a description. It was with lipid data, where you're comparing the classification of the head groups. And it looked to me like the collision cross-section and the mass ranges for your 95% confidence intervals were pretty similar between each of the classes. So how are you able to tell it's a PC versus a sphingomyelin versus?
Yeah, that's a great point, and that it would be where I would say, well, we're not going to rely only on the mass to charge ratio or a CCS. We will also be relying on the tandem mass spectra. Whether we can deconvolve all of that in terms of how these fall on top of each other, maybe not. I think it remains to be seen, and this also is drift tube eye mobility and higher resolution eye mobility measurements may get better separation, not necessarily in the CCS versus mass to charge ratio, but in overall measurement resolution.
And any speculation on what the ones outside the 95% confidence are? Are they just artifact in source fragment?
Yeah, we have not looked at the MSMS spectra yet that we've generated on the eye mobility instrument. So I wouldn't speculate as to what those might be yet.
Okay. Other questions?
I think there's a couple in the chat.
Yeah, I came back on. I was wondering just about this too, because there's also, fluorine shows up in some lipids, right, for fluorination, which would change the CCS value. That's not likely what's going in here, but that could be something like this could find those sort of outliers where you get some of that potential incorporation.
I couldn't quite hear all of that, Tim.
Oh, you couldn't hear it?
Not as well as I would have liked. Let me turn my speaker volume up here for a second.
Well, I probably didn't speak up. My wife always says I mumble. So
Yeah.
I was just saying that one of the structure, if you change like a fluorine or you change something in the structure, like one of the fatty acyl chains, that'll change the CCS value dramatically. So I don't know if any of that could come up. These are probably from positive ion, but since it's a sphingomyelin, but
Yeah.
those kinds of things could be cool with these kinds of graphs that could pull those out very easily that still may be sphingomyelin, but may have a fatty acyl chain that's changed somehow, that's changed in size.
Yeah. I think it would have to change in a way that does not alter the overall molecular composition such that you still get a putative match based on the mass to charge ratio.
Right.
I don't know if Shusao or Giancarlo want to unmute and ask. I have a couple questions in the chat.
I'm not sure what I'm asking actually, but adding the different dimensions, wouldn't you also need to weigh those dimensions accordingly to get an accurate probability of identification? So an MS2 probably weighs less than MS1 and retention time and so on in terms of how selective they can be. And if you add all together, do we also need to add their different errors and biases?
So in my mind, that's considered, so the tolerances or the errors that one allows in any dimension, it's up to the user. Here in this example, we chose a MS2 cosine similarity score of 750. Maybe you would like to raise that to 800, 850, 900. Similar to mass measurement error tolerances or CCS tolerances, I kind of view it coming back to the Hoberman sphere again, where this time if you consider the sphere as the multi-dimensional tolerance around the data point, you can make that as tight as you want, or you can expand it to be as loose as you want. Tighter multi-dimensional tolerances mean you may just identify fewer things at a higher probability. If you expand it, you might identify many more things at a much lower probability. So it's up to you to determine which tolerances you would like to use. And at least for how we've calculated and thought of probability here, I don't know if I would put any weights on it. I like the idea of applying the tolerances around each individual dimension, and you don't get a hit unless you get a molecule that matches within tolerance every single multidimensional property. And so if we're considering, say, the 4-dimensional aggregate here, and you get a hit on 3 dimensions, but your MS2 spectra sucks, well, that's not identification because you did not satisfy all 4 criteria. That makes sense.
But you could also anticipate a future where you're basing this on your 10,000 set of real world data rather than 2,000, cherry picked molecules.
Yeah.
That you can say, CCS is relatively more informative for something like a lipid that's a very, elongated structure, whereas it's less informative for an amino acid that's very clustered.
Yeah, indeed. Yeah. So again, tailoring the tolerances that one uses per molecular class. I mentioned that in the summary here. It would be nice to look at natural products and anthropogenic molecules as well, because they all tend to have certain structural characteristics that might not be captured in just primary metabolism, for example.
And there were some people who were using neutral loss, and so for the exposure compounds, and so many of them have chlorine and bromine. That's another piece of data that would be very useful.
Oh yeah. That's a great point. We could go to 5 dimensions and include a neutral loss aspect here. That would be pretty cool to look at.
Oh, I can do 6 dimensions and introduce NMR data.
Yeah. What? I'm not. Yeah, nothing against NMR, but I would prefer to have it all in a single integrated instrument. So if we could do that, if we could have all of those properties that we just measured and talked about, and then have NMR integrated, then yeah, I'm good for that. I just don't want to cut fractions and do that. I think of two concepts with metabolite identification. One is the high throughput stuff that we do, matching to libraries, and then there's the natural products approach where you cut fractions and you definitively get your structure that way. And what I'm referring to in this high throughput mechanism, they're probably more like identification hypotheses rather than definitive identifications. And so for a definitive ID, crystallography, micro electron diffraction, NMR, those are, I think, the way to go.
Any other questions?
Do you think that this can be a way to map all of the chemical space based on given measurable properties? That at some point we can actually pinpoint in space where a given unknown, given properties, where does it lie in terms of its similarity to neighboring spaces? Kind of like a smart NMR, which is a tool that came out a while ago that can visualize a 3D sphere of chemical space.
I think it's possible, but in terms of actually visualizing it, we're still going to be limited to 3 dimensions or combinations of 3 dimensions. So if you had a 10-dimensional measurement, you can only actually visualize 3 of those dimensions and be able to interpret it as a human being. So I would answer yes, but I would say you've got that caveat of what you can actually interpret as a human. Either visualizing the native dimensions or doing some sort of data dimension reduction through like a PCA or something. That might be a way to go about it.
Tom, may I make a quick suggestion here? Your selection of 2,000 compounds was probably random. So when we look at the initial serum DB from Wisher lab, which is only about 2,000, 3,000 metabolites, we found that is actually very good representation of what we see in the experimental data in plasma or serum. You can probably just calculate that 2,000, 3,000 compounds, and that will be very useful as reference for plasma serum analysis. Yeah, I would love to do that. This was a fairly recent data extraction and calculation, and I'll be working with David's lab to fine tune the actual composition of the molecules that we look at. And I'd like to go from 2,000
10,000, 50, 100, and just see how the data changes. That's what, that's what the proteomics folks do all the time because they have it relatively easy. They've got sequence, they've got Xtandem, they've got all these nice algorithms that, generate the fairly confidently identified peptides and allow them to do all these data games that we tend to struggle with because we don't actually have the experimental data for every molecule that we can measure. And not that proteomics measures every single peptide that's present, but they forget, I think, how easy, relatively easy it is.
And they've got a fixed library as well. It's a vocabulary of 20 letters as opposed to.
Yeah. And constrained to, a polymer with amide bonds and yeah.
Okay, with that, I think we'll say thank you again, Tom. Great presentation as always, and very